We've got a really lovely lineup of people here today.
Yes, so obviously Bridget and me, Henrietta from the Land Gardeners, and then wonderful Jeff Allen from Microbes. He has been making probiotics for the home and the soil for the last 10 years and is so fascinating.
And actually this year, as you know, some of you will know, we are making microbial compost. And we realized after a while there was so much muck coming out of the yards from the horses and from the cows after winter, we weren't able to process it all quickly. So then we turned to Andrew, who's an absolute expert on fermenting waste, which means it preserves all the goodness. And he's also a Nuffield scholar who's just returned, so he's going to fill you in on all things fermenting.
And finally, but not least, we have our piping wizard from Northumberland, Tom Fairfax, of a Mindrum farm, and there is nothing he is not making on his farm and testing.
Okay, so what's happening now? What are we doing, Henry? We are it's really exciting. The last 18 months we have been based up at Althorpe. Some of you may know of us when we were producing organic cut flowers on a horticultural scale.
Yes, we did that for over 10 years, and Bridget and I were obsessed with soil health. That was really actually why we did it. We started travelling to discover how we could really look after our soils, and that was gardening, that was growing flowers. And we ended up in, we did the Elaine Ingham, we ended up in Austria with a really incredible couple called Angelika Lubka and Urs Hildebrand. And their parents had worked with Pfeiffer, who in turn had worked with Steiner. And they developed this method of composting. It's an aerobic method of composting. So it's all about getting oxygen in for the microbes. And Bridget and I were doing it at a horticultural scale when Ben Raskin from the Soil Association came and said, "What are you up to? You need to do an innovative farmers trials," which we did. And soon we had jumped the garden wall and started making it at a farm scale.
So we were very lucky to go up and get some nice barns up at Althorpe Estate. Hopefully, yeah. And here we are. We've started making this aerobic composting method up there. And over the last 18 months, we've been trialling it on 250 acres. And then to our astonishment and wonder and gratitude, the estate decided to turn the rest of the estate that's in hand up there, which is 2,800 acres, organic. And they're hoping to try and use amendments that we are going to be making from ferments to vermicast to our aerobic composting method to try and feed the land and stimulate the microlife.
And that's really what we realised, is that we needed to trial it properly. We needed really to get some rigorous results. We've seen it at a horticultural level. We managed to increase our carbon levels from 5% to 12% over 10 years where we were growing. But we really need to do that on a farm, and we need to share others and what they're doing. So that's what's really exciting now. And if any of you were interested, please just please come and talk to us. Please go onto our website, and we've got a newsletter, and we're going to be sharing all those results from up there.
The brilliant thing about what we're doing is that it's going to be all open source. So any successes we'll share, and all our failures as well, which we've had many, and we'll have many more. But what we're hoping to do is create a toolkit for farmers and growers, so that they hopefully don't have to go through all the silly nonsense that we're going through, and that there'll be methods that they can adopt onto their farms. And it might be, composting might work, but also it might be sort of microbial teas, or it might be vermicast, and you'll be able to come. Hopefully there'll be lots of times where you'll be able to come and visit us, or there'll be newsletters going out of what we're doing.
So really it's about finding the most efficient and beneficial way of recycling your waste on farms, so that you don't use chemicals.
And as you can see down here, there's lots of fun and games going on. On the left we're making windrows, low windrows, which are very aerobic. And also, it's an amazing thing. The microbes seem to break down the waste in a pretty short time, between sort of 6 to 8 weeks. So, and it's so fine that we're able to inoculate seed with it. And amazingly, we're even managing to get it through a direct drill, and not clog it up, thankfully. So that's pretty exciting. So the whole idea around that is that all the microlife that's in the compost is coating the seed, and when that seed goes into the ground, it's got that it's sort of in a little nest or a little egg of goodness that will really help it start its life. Because as we know, plants come from a seed. You need a good seed. That seed needs to be strong. It needs to have a good start in life, a little bit like your children. And then you grow resilient plants that will be more resilient to pests and diseases. We've seen this on a horticultural scale, and we're really hoping to see how we can scale this up on a farm scale.
So the other method of spreading it is compost teas and compost extracts, and this was devised by Tom, who's head of our composting, who is over there. And if any of you want to learn how to make this, so that it's actually very simple. It's an IBC, it's a pump, it's a nozzle that we actually got from New Zealand. But Tom is your man and can talk you through how you could make it.
It's not going to do 2,800 acres, you can imagine. So we are looking at other ways, and people like Tom Fairfax here is developing different ways of spraying for larger areas. This is what the brilliant thing about what we're doing is, that this whole collaboration of ideas and sharing. None of this has been done before. I mean, all of this has been done before, but a lot of it hasn't been shared. So we're trying to get and bring all the knowledge and into one place, and hopefully get it out to you guys.
So we've been looking at vermicast as well, and bokashi, and also trying to make our piles actually more fungal. So that's a really interesting thing, whether we put them first through a bokashi and then we put them through aerobic, where we put them through aerobic and then they go, then we put the worms in them. It's just basically just making lots of potions and having quite a lot of fun.
And so another very exciting thing about what's happening at Althorpe is that Nicole Masters, who hopefully you're all going to see, and some of you actually have been with her the last 2 days at Althorpe. We've had an amazing 2 days learning from her. You must go and hear her. She's great. Also another fellow Kiwi. Now she has very kindly agreed to sort of oversee this whole project at Althorpe. And another very exciting thing is, many of you will know Garth Clark, who is an experienced farm manager. He's been made head of agroecology, so we've got him to work with. And Abby Rose of Soil Mentor has been helping us as well. So we're in very good hands. And another thing that's we're casting out around England to see all the people that are doing exciting, interesting things. We'd love to hear from any of you. The other thing is we're looking internationally. This is in Zimbabwe. We're working with someone called Claire Griffith, who learnt our composting method and has taken it way beyond what we could have ever imagined. She's built labs, and what she's doing is she's making compost and then inoculating that compost with the microlife that grows under the specific trees that she's wanting to grow. So they've got big avocado farms, they're planting lots of native trees, all of which are coming with compost that have been inoculated with beneficial microbes that they like to grow in, and they're getting incredible results. So and there's people that we're working with in Sweden, in New Zealand, Australia, and America, because there's really exciting things happening over there as well. Oh, look out.
And then, of course, it's about microbes. Just as our health is about the health of the microbes in our gut and we're all trying not to use antibiotics. So with the soil, we're looking at it in a similar way and thinking, how can we feed those microbes and get them really working in the soil? And the other thing that really weaves into that is energy. And unfortunately, Jane Scotter couldn't come. She's at Fern Verrow at Heckville and at Heckville Place, and she is phenomenal. So if you don't know her, really, if you can visit there or try to follow her, she is she knows everything there is to know about biodynamic farming. And we're very grateful now that we've got a biodynamic gardener who up at Althorpe, and he is also helping to share his knowledge, because so much of this is actually about energy.
One thing that we are also going to do in 2025, we've been asked to do an exhibition on soil at Somerset House in London, and we were asked to do a proposal for this. And I actually have to say, when we'd finished the proposal, the panel of curators looked at us with these big eyes and blank faces, because what we'd actually done, they'd expected that we were going to show them lots of pictures of soil and creatures and things. What we'd done is we'd started off in space, and we had some astronauts coming down to Earth, and then coming right down into the earth, right under the layer of soil, and then going back up again through the grasses and the flowers and the trees, and up into space again. Because what we've all done is we've siloed soil, grasses, flowers, trees, and we don't, we've forgotten that the energy that flows through all of them is really the key. I mean, what's happening with the moons up there is really affecting the microlife. It stimulates the microlife in the soil. And if we can work with those energy patterns, like everybody used to 200 years ago, I mean, you wouldn't have planted your wheat without seeing where the moon was in conjunction with the stars. And I think that's all of this is one of the other things that we need to start thinking about when it comes to soil and soil health.
That's why we also need to have scientists working with us, because yes, otherwise we might lose some people. And of course we need scientific rigour, and that's going to be the great thing about this soil lab up at Althorpe, is that we are going to it's going to be university led, which is really exciting. So
yeah, I mean, I don't know, has anyone here used any biodynamic preparations? Okay.
Great,
yeah,
that's great.
That's great. And have you had positive results? Oh, yep, there's lots of thumbs up.
Yes, there we are. Look at that. Exactly.
I mean, honestly, when I started bearing horns on our farm in New Zealand 20 years ago, I can tell you all the shepherds and the managers thought I was completely kooky. But it's amazing, and I didn't even really know what I was doing, and I didn't understand it's all about energy at that point. What I started growing and what the difference we had in the fields was incredible. But I'm sure something like biodynamics, the science will catch up with it at some point. And I think that's us. Over to the other lot. Thank you.
Thank you.
Thank you for that. I hope you don't mind. I'm going to sit down for this. Yeah. So, first slide. Got that coming up? Yeah. Oh God. Oh yes, please. Yeah, thank you. Oh. Yeah, we're Microbz. We come out of Wiltshire. Like they said, we've been working with microbial cultures for about 12 years now. I've been traveling around the world, where I came across them in Australia and in Austria. And we still have two partners that we work with, one in Spain and one in Germany. So you'll see some of their work in this slideshow. But I'd like to start at the beginning. Oops. So this is the timeline of us on the planet. And basically the planet started here about 4.5 billion years ago. And life started showing up about 4 billion years ago. And the form of life at that stage was microbial. So the scientists tell me, and I wasn't there then, so I don't know, but that the scientists say that the world was covered by a mat of microbes. And this mat of microbes, from that, all life came. And that's the progression of life. And we are right at the end there in about just a tiny part of it. But the purpose of this slide is to recognize that that original microbial culture that covered the earth is still there. This can be seen in some places like in Yellowstone National Park and other places around the world where you can go and that microbial mat still exists. So what we do in order to harness beneficial microbes is to go and basically extract them from the soil. And we've done that in the UK, we've done that in Spain, we've done that in China, we've done that in Africa. And when you do that and you extract the microbes, you get almost identical cultures. So this is a breakdown of the microbial cultures that you'll get from soil microbes. And you'll see that in this particular slide, this is 2 different times of the year in 2 different locations in the UK. And you'll see that it comes out within 2% of the same number of microbes in the same proportions. And so if you went to China, as we have done, and dig a hole and bring it back and analyze it, you will get the same answer. So that microbial mat is still there from which all life came from. So what we're doing when we're using microbes is basically we're reconnecting back to that source. We're reconnecting to where basically all life came from. So this is basically, so microbes, is this our presentation? I thought this one was out, but it's here. So here we go. So microbes, what microbes are really good at is closing the cycle of life. We as, what we've done on the planet, we're not very good at closing the cycle. So the microbes close the cycle. So it doesn't matter where you introduce them in the cycle. Like some dairy farmers, for instance, we've worked with, what they want to do is to stop their slurry smelling. So if you add microbes to a slurry pit, the first thing that happens is the smell goes. And then you know you've changed the process from putrefaction to fermentation, which is what microbes do. And then, of course, you put that on the land, and then the animals eat that, and, round it goes in that cycle. So after a while, it's much easier for the farmer just to add it to the feed rather than to put it in the slurry. And once you start that whole cycle, then that health passes all the way through. Looking forward to the next slide, whatever it is. Oh, yeah. So let's leave it on that. So here thank you. We've talked about microbes, and you'll hear more about how beneficial they are. But of course, one of the problems is how do you get them out into the farm? How do you get them into the soil? So here you can obviously spray them directly onto the soil. You can spray them as a foliar feed. You can use them with a subsoiler, injecting them into the soil. Or you can add to a compost as an accelerant, and then, which I'm sure you'll hear lots more about. Or there's a way of rotivating a cover crop and spraying that. So we'll go through those, I hope. So here we are. This was a trial we did in the New Forest. We built a subsoiler, and at the back of the subsoiler, we put pipes down with 2 nozzles, and then mounted the tanks on there, and we subsoiled some land. The dosage was about 150 litres per hectare, of which 100 litres was microbes, 50 litres was water. And then the result of that is this. Oh, there's the tractor moving. That's great. That worked. So here's the control one. Obviously compaction, claggy soil, 5 slow-moving juvenile earthworms, So that's what the control one that wasn't subsoiled and didn't get any microbes. So this was the result having from the subsoiling and injecting with the microbes. So obviously the compaction had gone, the soil was crummy, and there were 15 active earthworms, including 2 adults. And all that was achieved within 3 months of subsoiling with microbes. Right, moving on to the next one. It's when you think about what we do quite often with our compost, we go and collect it out of the field, we harvest it, we forage it, bring it out, put it in a clamp or a windrow, and then we do all this sort of whatever process we do. Then we take it out, put it in a sprayer of some kind, and go and spread it back on the land. So this is our partners in Germany. What they're trying to do is short circuit that whole process. So they get a rotavator, and they've actually designed their own now, which spins at quite a good speed, so higher than most. So then they rotivate the cover crop, usually just touching the soil. So you mulch it up, and at the same time, you're injecting microbes into it. So then you go over your field, you set up that mulch, which has now got microbes in it, so it starts to ferment, it starts to compost. And usually what they do now is they run it again a few weeks later, and they'll set it where they go into like 1 centimeter of soil and run that again, and at the same time, more microbes. In some cases, they're drilling straight into that. So you get this that microbial mulch on the top, so all the microbes, so that is now already composting as it sits in the field. So, and obviously the root layer underneath is still there, which allows for the permeability of the whole process. And so you don't have, bare soil at any time. It's always, you're adding the cover crop in, and it's integrating in with the soil. So they're getting really good results with that. So this is another example that came out of the German experiment. So this is a piece of land that's been harvested, and it has the 2 pipes that are sunk about 1 inch into the soil. Now the one on the right, about 3 hours beforehand, that piece of land was sprayed with microbial cultures. And then water was added into both of those. And you can see the obvious results. The one on the left, the water's still in there after half an hour. It's not getting absorbed into the soil. Obviously, the one on the right is now, all the water's drained out, it's now in the soil. So that's an experiment and about how beneficial microbes in the soil makes it much more porous and helps for the water retention, which is becoming more and more important now. The rain is getting heavier, it's not coming as often, all those things. So this is an example of that. Obviously the maize on the right has been treated with microbes, manure, and sprayed as a foliar feed, and the one on the left, and this was at a time of drought, so it's not saying you're going to have amazing crops each time you have microbes, but you get much better water retention, much better drought resistance if you put microbes on there. I think we're coming to the end. We got time off for bad buttons. Oops. And
God, is there any technical department around?
yeah. Wow.
I think the battery's
The battery's gone, yeah.
yeah.
Just this
One before?
one before, yeah. So this is composting. This is composting in Spain, done, and this. I'm sure Andrew will talk a lot more about this. You don't need to turn it. You just spray the microbes on there. We put microbes with other additives on there as well. Phosphoric acid. Microbes really love that sort of stuff, and it helps break it down. The main thing is if you do it, this method is no turning. And now this is a windrow that's all been treated with microbes. That's the size they use it. This is in Spain. Once again, you only need to turn it 2 or 3 times maximum. The whole process goes through in about 3 months instead of the normal 6 months it used to be. And they recommend treating each crop with about 10 tons of compost per hectare every 2 years. And the cost is about £40 a ton. And I think that...
That's good.
Yes, that's... Thank you. Soil-based microbes, the ultimate renewable resource.
Very happy to give you that.
Thank you. And there's how to contact you.
Yeah.
Right, I shall stand. So hi, I'm Andrew, and I've just been asked just to touch a little bit on my first stint as a Nuffield scholar and my first travel. I was fortunate enough to go to a number of different countries: Canada, New Zealand, Kenya, Chile, and sort of the take-home message from me in the conversation that we had when we spoke earlier this week was that several of the New Zealanders that I met said that they were about 5 years behind Northern Europe. In fact, they actually looked to Europe to see what was coming next. Chile was probably another 5-10 years behind New Zealand, would make them maybe 10 years behind us. Whereas Kenya, they were probably 50, 60, maybe even 70 years behind the UK. In fact, I would say they're so far behind, they're arguably ahead. I think the way in which they were reusing their resources, they were far more resourceful than what we are in the UK. And I wonder whether that's because they don't have the access to, and Nicole mentioned it, I'm going to use her saying, is you speak to an agronomist about a problem and they will recommend 250 kilograms of nitrogen as a solution. That's not as easily achieved in parts of Africa. So, as I said, far more resourceful. And it's something that I'm happy to talk about, as I know there's a few other Nuffielders here that would be more than happy to talk to you about their travels so far. So, as you can probably guess, I'm looking at the management of organic manure. Why is that relevant? Why is that important? Really, really simply, hopefully everybody that's here today can recognise themselves in one of those pictures. We've probably got arable farmers, dairy, beef, horticulture, flower farmers, horses. The common denominator? Soil. Soil is the foundation upon which every single farming enterprise is built. It doesn't matter what type of farmer you are, soil is the ultimate resource that you have and will be the success or potential failures of those farming enterprises. So as we know, soil traditionally has always been a medium in which we grow crops. So we plant a seed, the seed germinates, we force feed it with synthetic fertilizers. We now know that the soil is alive, full of living organisms, and just like us, what do we need to eat? We need carbon, carbohydrates, we need proteins, nitrogen, also a little bit of water, a little bit of air also helps. But we know that the soil is alive, and therefore living organisms need to be fed. So when it comes to feeding them, what do? We look to organic manures. There are, of course, other ways. You can talk about green manures, cover crops. But organic manures is what I want to touch on specifically. And rather than sort of smaller scale stuff, we want to be looking at larger scales. So on farm, this sort of site, I would imagine, is fairly typical. And I'm hoping that if I press it next, we'll see a little video. Nope, not today. It's not just you, it is me as well. It is. So this is a typical site on farm, and what we can see here is an awful lot of steam rising from that muck heap. So within a couple of weeks of being cleaned out of the sheds, put into a store, that muck heap is generating a huge amount of heat and releasing an awful lot of energy. So we talk about feeding the soil. We want to be feeding the soil with energy, with carbohydrates, with proteins. We're losing an awful lot of them. You can quite literally see the losses that you're experiencing on farm through the steam, volatilizing. That would include carbon, potentially a little bit of methane, some nitrous oxide, but also the leaching. So you'll see leachate running down into the water courses, potentially causing pollution, wider pollution, and having a negative effect on the environment. So what can we do to prevent the loss of these nutrients, which we all know exactly how expensive these nutrients are to buy these days? So what can we do? Something that we're already doing on farm, highly likely, is fermentation. quick question, who's making silage on farm? We've got a few hands, that's good. Why are you guys making silage? You're making silage to retain the excess nutrients that you're growing in the spring and the summer to feed to your cows in the winter months. What would happen if you were to take that same silage and put it in the corner of a field in a heap and leave it exposed to the elements for a period of time? Would you then feed that to your cattle 6 months later? I don't think so. Would you? No, probably not. So instead we ferment. So putting that organic manure, organic matter into a fermentation state. So in this instance, as we can see, we've got an ag bag at the bottom and we've got a bokashi heap at the top, which has been covered with black plastic and straw to keep it there intact. So fermentation of the organic matter will retain those nutrients. Those microbes that we're adding into the bedding during the housing period, which hopefully will appear now. There we go. Again, this should have been a video, but it's probably not going to play. The application of those microorganisms that Jeff's been talking about and similar microbes applied onto the bedding during that house period will reduce the smells of the sheds, will help reduce disease pressure, will add value in regards to animal health and welfare. But those microbes will also start that fermentation process. So you're not going to be smelling the ammonia in the sheds. What does ammonia contain? It contains an awful lot of nitrogen. So you're retaining more nitrogen even in the cubicles or in the bedding areas. And those microbes then will start to ferment that organic matter. And that fermentation process is not only retaining the nutrients, but it's also starting that breakdown process. So those microbes that Elaine Ingham talks about at the bottom of that soil food web, they're the microbes that we're using. They're the ones that are going into that organic matter first to start that breakdown, to start that decomposition. But it's going to be through fermentation. Oh, look, we do have video. Here we go. Look at that, and as if by magic. So quite simply, in this instance, we're spraying the microbes on. This instance, we're using a watering can. A knapsack sprayer works really well as well. There's also people that have established and set up misting systems throughout their shed to apply the microbes, as well as it being applied onto the straw, as the straw is being blown into the sheds as well. So a number of different ways in which you can apply. And then once you've applied, we're going to go to the next slide in a second. Once it's been applied, it's really simple. The cows go out, the muck comes out, you put it into a heap, you seal it, you cover it, whether that's with a black plastic sheet. We've got people that are using straw, blowing straw on top of it. There's a number of different ways in which you can cover it. That covering does two things. Firstly, it keeps the rain off. If you've got rain falling on your muck heap, what are you going to be losing more of? More leachate, more soluble nutrients, more value. It also maintains that anaerobic environment. That anaerobic environment is important to the fermentation process. The question we always get asked is, well, anaerobic is bad. And if you've got anaerobic soils, I would say absolutely. The microbes that we're using in this instance are what we call facultative, which means they can switch between aerobic and anaerobic processes really very quickly. So as soon as this is then applied onto the soil at a later date, those microbes that have been working anaerobically to ferment the organic matter will switch to becoming aerobic and working aerobically within your soils. So not only are you providing a nice bit of food, nice bit of carbon for the microbes, the living organisms that are within the soil already, so earthworms, beetles, spiders, nematodes, protozoa, but you're also providing additional microorganisms, beneficial microbes to the soil as well. And by maintaining them and by providing them with a food source as well as the microbes, they're going to hang around a little bit longer while that soil recovers and builds its own natural soil health and fertility. So I think I've rushed through that pretty quickly. I think you've got the grasp of it. For anybody that's interested, I have got one I prepared earlier. Literally just at the bottom next to the soil medic tent, there is a actual bokashi heap that you guys can come have a look at. You can do what I do and get your hands into it as well. We haven't got any paper towels to wipe your hands, so be prepared for muddy jeans, but you won't have smelly hands, as Jeff pointed out earlier. So that's me in a nutshell. Any questions, come and speak to us afterwards, and I look forward to telling you all more. Thank you.
Hello, everyone. You've heard from the steely-eyed professionals. I'm now the rank amateur. Over the next 10 minutes or so, I'm going to give you a quick a chat through where I come from, what I'm doing, and my context. how were farming using a lot of this, and one or two of the outcomes. There's a theme to which I will refer again, which is that a fool with a tool is still a fool. And so I'm hoping that this will work. My name's Tom Fairfax. I farm in Northumberland. We're a mixed farm on the side of the Cheviot Hills. It's a lovely place. The ground's pretty thin. And the margins are pretty thin too, and getting thinner in a conventional context. The thing that I've always been fairly minded about the place and about what's living on it with us, but we have been in a conventional farming model. The first thing that's been vexing me for some time is when I was a kid, we had did anybody remember a thing called a class matador combine? It was, I think, an 8-foot cutting bar, you know. It was rare that we would finish our harvest before the last week in November, by which time it was an interesting state of black. But the peaks of the harvest in the trailer were always red with ladybirds. And I haven't seen that for years. Well, actually I have now, and so we've gone into, over the last 5 years, we've gone into a more regenerative model. We've then gone on into an organic model. So we're now in a regenerative organic model, and we're pretty soil focused and microbe focused. Done quite a lot of retraining, both myself and my team. And those of you with teams who have been around for 40 years or 50 years and several generations will know that that's sometimes quite challenging. Anyway. Effectively, we're light sandy loam, lots of whinstone. Our underlying rock is the same thing that they built Hadrian's Wall on, and it's pretty thin. We've actually shifted not just into this biological model. It's been an entire shift that I wasn't really expecting in terms of the operational model that we're having to deploy. And we're learning a lot all the time. For me, the microbes and the biology is important, but it's the information model that I think is really interested me. We've moved from a conventional system where we were directed and we imposed an operating model on the ground. We've moved to a regenerative model where it's responsive, it's agile, it's contested. When we do something, if we move to the left, the place in which we're operating is going to move around us. We're not just watching it, it's watching us at the same time. Everything we do, or in fact everything we might not do, which makes it really shaggy and complicated, is going to have an impact. And we've got to read that. And so for me, the real challenge, and actually the really exciting thing that makes it so much fun, is that we're listening. We're now beginning to talk to the place and having to listen to the place and listen to what's going on. And so it's that active model. I got really frustrated with looking at soil samples every 3 years, and they didn't actually tell me anything that I could use. So from my point of view, I see my place as a rather bossy aunt. Has anybody here got a bossy aunt? Is anybody here a bossy aunt? I bet you have. Yeah. You know, actually, I have this lovely, wonderful aunt who I got really cross with my mother. Every time she's got something important to say to my wife and I, she talks to my wife. I get crosser and crosser, think, well, why don't you bloody talk to me? Come on, I'm a grown up now. And I was whinging to my aunt, and she said, look, Tom, you've got to remember that to every mother, her son will always be 7 years old. But she met your wife when she was a grown up. Who the hell do you think she's going to talk to? So anyway, the framework that I'm using as my prime framework, most of you will be familiar with this, is ecological succession. Where I my operating model, the things I'm trying to produce are generally mid-successional. Productive grass, we've got sheep, we've got a suckler herd, and we've got some arable. We want to be about mid-successional for cereals and for productive grassland. Having been in a conventional model for 30 or 40 years, most of my ground, to my surprise, when I started looking at it, is like the side of a volcano. There's absolutely bugger all in the ground, alive. And that's because we've created conditions. And actually, let's face it, the conventional model was there for a reason. Most of us here have parents who grew up with ration cards because this country nearly ran out of food. And so we've been focused on producing calories. And the system has worked bloody well. And in order to do that systemically, what we've done is we've basically wiped out the soil and made it into a dumb matrix. And that has worked pretty well. As most of us in this room are probably aware, there's been some side effects. And a lot of those side effects were just coming on. So my task is to get from the left-hand edge of that succession into the middle. And so what I'm doing, I'm using the microbes to move myself up there. There's lots of other spectra that we also use. There's things that indicate, what's growing in the ground. Have we got, I love that thing, when somebody comes and whinges to you that they've got arable weeds in their organic second wheat. You don't need to actually look at the soil to know what the problem is. Because if you've got arable weeds, which are pioneer species, most of them, you've got nothing living in your soil. And if you had mid-successional soil, your arable weeds wouldn't be germinating, or few of them would be. And this is the magic. The tool I'm using to navigate myself up is the soil food web. I'm doing a lot of training and I'm doing a lot of the Elaningham stuff, amongst other things. There's lots and lots of absolutely brilliant paradigms out there and doctrinal models. And the lovely thing is it's like a sweetie shop. There's lots of really good tools. But because everybody here is on a different bit of ground, we have different context. So we need to use different tools. But we've got an astonishing toolkit that we can pick from. But we've just got to know what's going on, what exam question we're trying to solve today. Because the other lovely thing about the biology is that what it's doing today might not be the same as what it's going to be doing tomorrow. Because it changes, it lives. When we put when we put a drench on the ground, we change the ground, we change the rules. And depending on what, if you're very acidic or you're very, if you're a very high pH, that biology will have a different effect. So we've got to be listening to it. Now, the soil food web, sorry. I'm assuming that most people here will be familiar with the soil food web. It's this whole matrix of things eating other things. And the fact that as you get bigger, you spill lots of nutrients. And it's this nutrient cycling, pumping the nutrients up this soil food web that makes them available to plants. What I'm doing on my farm, I'm using the food web to mine the nutrients in the ground. Most of us who have been conventional farming have got around about 90 years of nutrients tied up in our soils that are unavailable unless you can make them available using microbes. And nature has this wonderful model whereby the bottom of the food web will mine those nutrients and make them available to what we're trying to produce. The challenge is, if the bacteria and fungi at the bottom of the food web are eating and producing these nutrients and are just on their own, you've got a whole lot of nutritious microbes sitting in the soil. And there are ways that the system can get it. I know you'll all be aware of rhizophagy, this lovely thing that plants aren't vegetarians. I think Nicole Masters should be credited for that. There are ways that nature can get the nutrients out of the system, but really it's getting this food web, getting everything, eating everything else. And that's the exciting thing. So, how are we doing this on the ground? Firstly, I bow to the masters and the mistresses. These are the compost gurus. We make lots of compost. There's lots of different. We do bokashi. We do aerobic compost. We do large scale and small scale. I've got some Johnson Sioux going. The lovely thing is each one of these produces a different tool. They're all slightly different. And so long as you can see what you're doing, and I use a microscope every day. I have one on my desk, and so long as you can see what you're producing, see which tool is relevant, you can create the cocktail that helps you solve the problem. But you've also got to be looking at the ground. So because that exam question strategically might take 5 years to get your ground right, it's moving every day. So it's looking at what's on the ground, looking at the gap, looking at the tools, building the solution, putting it on, and then seeing if it's working. And being typically male, of course, it's really cool because it's quite quick gratification. You can see it happening pretty quickly. Hang on. We're trying to make most of our inputs on the farm. We're doing a lot of things. Anybody working with Korean natural farming? There's a lot of systems like that which are really cool. There is a huge amount of stuff on the ground that we can capture. We work a lot with lactobacillus, the bokashi. I love the blend of things. When we're making some protozoan infusion. Protozoa, absolutely key. I've got a real lack of protozoa on my farm in the soil. I've got quite a lot in the hedgerows, curiously. These are the predators that really release the nutrients. We were laughing about this earlier. I love the lactobacillus and the facultative anaerobes, Andrew's things, which are absolutely great. They are magical little bags of nutrients, and one of the big challenges when you're putting the predators, and this stuff is Shakespearean, it's cry havoc and let slip the dogs of war. Pumping protozoa onto your fields, they suddenly release all of these nutrients which are sitting there waiting to be dealt with. But if you're putting the lovely thing, if you're putting anaerobic, facultative anaerobes on, these things are absolutely stuffed full of nutrition, just waiting to be eaten. And because they're facultative anaerobes, it means that they really, they can exist in the aerobic soil environment that most of our target species, most of our target crops are really wanting. But they're struggling a bit. So the analogy I was using yesterday, which may be inappropriate, is actually probably entirely inappropriate. It's like breeding asthmatic pigs to feed lions. Asthmatic pigs are stuffed full of nutrients, but they are they're not terribly competitive in the sort of environment where you've got lions. But it's all this. It's these blends between all of these different tools to make sure that we're getting the action in the ground. Biochar. We've got light sandy loam. Sometimes it's hard to get the biology to lodge. I'm doing a little bit of work with biochar, really cool, inoculating it with the microbes, and it just allows it to lock in and start working. A lot of you will be working with biochar. It's really cool. Lots of R&D and experimentation. We're doing not just the inputs and the composting. We're working a lot with seed mixes, trying to different ways of actually managing the soil. We're trying to work a lot with green. I'm calling it a green canvas. I'm trying to get to organic nil-till, which of course is slightly eye-watering. and deeply hair-raising most of the time. And we're trying to create a low-green canvas with clover and forb mix, into which we can direct drill productive cereals, and on which we can graze sheep and cattle. We're using the livestock as engineers. We're trying to manage competition with height, light, and temperature. Real cock up last autumn. My theory for this is that you eat the clover hard with sheep just as it's getting cold. So the soil temperature is getting down to about 7 degrees. So the clover's starting to slow down. But you've still got about 3 degrees of wheat temperature. So the theory is you get the wheat in, the wheat gets ahead, and then you graze it a bit during the winter. And then in the spring, of course, the wheat then starts growing faster before the clover gets away. So it then starts competing with the clover with light. But when you then harvest it, you've got a clover lay underneath. And it was going really well. And then we in Northumberland, we got that warm thing in November. So it was looking perfectly. And I made that mistake, and we've all done it, of feeling smug. You know, you think you've cracked it. And yeah, anyway. It's actually looking pretty hair-raising now. It's now looking better. It was looking really, I thought it might be whole crop silage about 2 weeks ago. But it surged away again. So lots of things like this, trying to make it work and trying to get into a system. The other things that you'll all be doing, lots of really exciting tools. I'm not going to go through this slide in detail. These are all things that we all have available to us, tools to use. The challenge is this information model. There's a lot of information, there's a lot of science out there, there's a lot of really good stuff. But if it's going to be useful to us, information has got to be timely, it's got to be relevant, and most specifically, it's got to be actionable. And so what I'm really working with on a day-to-day basis, and this is one of the things on the land garden side, we've got a whole lot of little lenses and windows into my rather hair-raising information space. You know, whether it be looking at something, you know, where bats are flying at night, what grasses and what things are growing on the ground, looking into a microscope and seeing actually what's living in the ground, what aggregates are in the soil. You know, lots of talk about soil organic matter, but as most of us here know, soil organic matter is not necessarily soil organic matter. Is it necromass? Is it biomass? Is it microaggregates that have been processed by bacteria? Is it macroaggregates that have been stitched together by fungi? All of these things. And they tell an incredible story about the ground, and they give us the management information we need to actually drive this thing forward.
Thank you so much, Tom. Bravo, bravo, bravo. And thank you to Tom and Andrew and Jeff. I mean, it's brilliant. They've opened lots of doors and windows to you. Sorry, no time for questions.
But if you want to come and ask us any questions, we are now going straight to the Soil Medic tent, and you can come and ask us and see all of Tom's tricks.
Thank you so much for coming.